Hi everyone, I'm Professor Fan, and today I'll be doing a live coding session on a spatial transcriptomics exploratory data analysis. And let's see, to get things started, why don't I share my screen? I can just share my whole desktop, and I already have my RStudio session set up. Why don't we also just make a folder for organizational purposes? Let's see. What should I name it? I guess I will just call it Coding Nov 2022. And let's see. Why don't we create a new project in a new directory, the existing directory actually, to keep things organized. And okay, so this exploratory analysis is actually inspired by a question from a student when I was giving a talk at the Fred Hutch. So I was giving a talk about computational tools for spatial transcriptomics data analysis, and I was talking about a tool my lab recently developed called STdeconvolve, which is able to take these multi-cellular pixel resolution spatial transcriptomics data sets and recover cell type proportions in these spots along with cell type specific spatial expression profiles without reliance on an external single cell reference. So to learn more about STdeconvolve, I encourage you to check out the software, and there's also a video that you can check out as well. But one thing that I showed in the talk was that for these multicellular pixel resolution spatial transcriptomics data sets, sometimes the cell type proportions we infer look a little bit different from kind of the single cell resolution spatial transcriptomics data sets on the same tissue. So the student was asking whether, I guess, there was a potential lateral diffusion in terms of the mRNA capture, whether STdeconvolve could be combined with some kind of bioinformatics approaches for fixing this lateral diffusion issue. I think in particular, I mentioned this thing called SpotClean. Oops, SpotClean spatial transcriptomics. Yeah, so let's see. SpotClean is another bioinformatics tool that adjusts for. Let's see what this paper is calling spot swapping. But essentially, what happens is, I guess, the genes, rather than being captured at these specific spot locations, to kind of diffuse outwards. And and you can see that, this is a nice figure where you can see the human UMIs are detected in the mouse areas, and the mouse UMIs, oh, wow, it actually diffuses pretty far. Because, it's never a perfect capture. So so the question was whether we could do this type of cleaning prior to deconvolution to perhaps get at a better cell type specific spatial organizational profile. And so let's see what we can do. Okay. Oh, actually, I'm already here. Oops. To get started, let's first do GitHub. Oh, perfect. Let's first try to install. Let's try to install SpotClean and get things going. This is an older computer, so actually I forget what things I've installed here. Maybe I've already installed. Oh, good, I've installed DevTools. I'm gonna install the GitHub version, why not? Don't want to build the manuals. Control K. Oh no, this computer doesn't have my keyboard shortcuts set up. Oh no, it does. It's just the control and the command have been flipped. Oop. 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 Huh. Okay, all my keyboard shortcuts are a little bit messed up. Control D, Control A, Control Enter. Okay. Keyboard shortcuts are very helpful. Encourage all students to learn. Okay, let's see. Do I want to update things? I'm gonna pass. Okay. Okay, while that's installing, we'll also need a data set to play with. So why don't we work with the 10x Genomics Visium and a data set? Oops. Let's download a publicly available data set to play with. Oh my God, where do we find these data sets? Oh my God, just let me download the data set. Okay, data sets. And let's see, and the talk I was presenting on, it was a spatial transcriptomics data set of the mouse FFPE demo. Yeah, so why don't I just stick with that? Okay, it's fine. Yeah. Let's see. What is my area of research? It's bioinformatics. Okay. Wonder how many instances of me are in their databases. Anyways, okay, so let's view a quick summary of this data set just to remind myself, is this what I want? Yeah, okay, great. Yeah, so even from the histology, you can see this is half of the mouse cortex. We can see a nice hippocampus, dentate gyrus, even just based on this H&E stain. So we would kind of expect the cell type spatial organizational patterns of those cell types in particular to be quite similar. And let's see, okay, what data do I actually need to get this to run? Oh, looks like it's still installing. Assume. Okay, I need to actually install it and look at the vignette. Oh God, there's no way to actually look at the vignette. Fine, gonna look at the code then. Yeah, we can look at the code for the vignette. And let's see. So, it looks like you need a path to a folder of the CSV image and scale factor. Okay, so then why don't I create a data folder? Well, let's start downloading some things into it. Yeah, I'm just gonna download the filtered. They have a whole folder. Do I need the H5 or do I need the CSV files? So, oh my God, it requires, okay, I am not updating R right now. Okay, let's see. Okay. Step back. Let's see what we can do here. I really don't want to upgrade my R right now. So I'm going to do a little cheating. Okay, let's see. CD desktop. I'm going to create a temp folder. Yeah. Okay, I'm going to go into my temp. Come on. Okay, and I'm going to go clone this. Perfect. And up. I'm going to Emacs the description. I'm just going to edit the description. Yeah, I'm going to cheat. It does not depend on this version of R. Everything that's bioconductor, they make you update to the latest version. So yeah, so now that I've changed the requirements, I should be able to install our package from folder. Okay. Yeah, okay. I'm going to comment this out. And this is, I'm going to install it from desktop, temp SpotClean, and this should work. Come on. Oh, holding my breath over here. Okay. Right. I was trying to figure out which files I should download. And so let's see. Yeah, so I can either use, I can either use the H5, it seems. Or these files. Okay, it looks like, okay, fine. They're looking for these files. Oh, cool. Okay. My little cheat here has looks like it worked. Double checking it can install, or it's installed and I can run it. Great, perfect. Okay, there is an H5 format. Okay, I can download either. Okay, so I'm going to just gonna download the raw. Okay, so I need the raw, and let's see, is there anything else? Let's go back to the vignette code. I don't want to build the whole vignette, so I'm just gonna read it. And okay, I'm gonna copy some of their code. Okay, so we are going to load our 10x data, which involves the path to the raw folder. And then there is also the slide. So we need the slide. And okay, so going to drop this folder. Should have just command lined it. Not familiar with using this thing. Where did it extract to? So many emojis. Okay, fine. I need to. I'm just gonna do this command line. what is it called? Where did it go? Okay, let's see. What are my downloads? Thought I would have. Huh. Don't know where it went. Well, okay, fine. I'm just gonna try again. Yeah, there it is. Okay, I'm gonna unzip it. You can tell I have a couple of data sets. Now I can untar it. Oh. Well, I guess where did I untar it to? Is the question. Oh, okay. Well, that must be it. Let's see. I'm gonna env it. Stop to my coding folder, to the data. Okay. Looks reasonable. And fine. What else do I need? I also need the path to the tissue position CSV, the image, and the scaling factor. That's probably in here. Oh, it's going to happen again. Okay, let's see. Is there a spatial folder? Fine, I will move the spatial folder to my data. Okay, that looks reasonable. So therefore the path is going to be data. Raw. Okay, let's see, does that work? And this is going to be data, spatial, the positions, the image. Do they want the high res or the low res? Okay, they want the low res. And data, spatial, JSON, okay. Well, something happened. So yeah, let's just copy from their tutorial and make a slide. We can visualize the slide. Oh, cool. And visualize a heat map of yeah, the first gene. Great. Okay. I'm just gonna double check some genes. Yeah, so this is a type of tissue that we work with quite frequently, so pretty confident in my knowledge of the genes and the expected spatial patterns. I'm just gonna check a few of my favorite genes. Yeah, I guess this is where the tissue is. Oh, there's a lot of weird stuff. Oh, weird. Yeah, you can kind of see where the tissue is. This is a Okay, let's try some other genes. Yeah, it's a lot messier than I expected in that, well, there's a lot of lateral diffusion, it seems like. There are counts of this inhibitory neuronal marker being detected even outside the tissue. Other fun genes that should be, so C17A7 excitatory neuron markers. Okay, yeah, this is a very highly expressed one. Okay, we can see where the cortex is. Oh, interesting. Okay. Okay, well, yeah, so we visualize the data now. I guess the whole point is to run this decontamination or cleaning of our spots. And I wonder how long this is going to take. It's going to take a while. Okay, well, fine. I guess, that's what it is. Let's see. In the meantime, why don't I make a R folder? I like to organize my code. What should we name this? I'm just going to call it SpotClean test. Guess it's busy running this, so it's not letting me do much. But fine. Okay, so, well, I guess it's going to take a while to run. And yeah, well, maybe then we'll just check back once it's done. Okay, well, this is going to take probably a lot longer than I expected. Okay, I'm actually going to start up a new RStudio session just so that we can maybe explore some other stuff in the meantime. Yeah, so the other part of this is the reason we're pursuing this is to see if it could work in conjunction with STdeconvolve. So maybe we should just try to run STdeconvolve and see what that looks like kind of with and without the SpotClean. Well, that could be fun. Well, that would be what we would do anyways if we were trying to evaluate and compare. And okay, so yeah, I am going to actually, I like this function, so I'm going to try to reuse it here. And let me double check it's what I think it is. I think it's just reading in the counts matrix. But this is the raw, unfiltered. I guess typically STdeconvolve would actually start with the filtered. So we do require the spots to be kind of actually on the tissue. But we can probably get rid of that by just removing the spots with very low reads anyway. Okay, let me double check. Now that's what exploration is. So, okay, I'm going to read in this matrix. Let's see what it looks like. Actually, even before I do that, maybe I'll check the dimensions. Sometimes the dimensions are too big and doing a head is really ugly. Let's see. Oh my God, why is this taking so long to just read in the matrix? This is a big matrix. Okay. Yeah. Yeah, okay, perfect. And let's see, in this case, position is just that that spatial position CSV. Well, actually, what's the dimensions of positions? Oh, okay. These are all the positions, including the spots that have like nothing. Ah, crap. I shouldn't have plotted all 6 dimensions. We actually only really want XY coordinates. Looking at it, there's 6 dimensions. There's probably other weird stuff in there. Oops. Okay, anyways. Yeah, okay. Oh, shoot. This is why it's always good to look at your data. Because this is wrong. Oh, that's better. Oh, I should have noticed. There's 4992 here, which should have been 4992 there. Yeah, so I checked. So in this case, I only care about the 5th and the 6th. Okay, so now double check. Yeah, that's better. And yeah, it's indeed every single spot. And okay, so now I'm just going to copy this tutorial here from STdeconvolve. Let's see, STdeconvolve. So let's see, we are going to clean the counts first to get rid of all the poor lowly detected genes, but then also the spots with like no counts. So I want to actually filter out for a min, the min lib size. Well, actually, fine. Let's see how it looks first. Okay, yeah, there's definitely a lot of genes per data set, and let's see, how does this look? Oh, okay, it's not stringent enough, I think. So let's see, how more stringent should we filter? Let's see, now we double check the minimum number of genes detected in a cell and the minimum number of reads per gene. Minimum number of cells gene must be detected. Okay, I'm just gonna filter quite a bit. See what this looks like. Okay, maybe that was a little rough. I'm trying to remove a lot of the cells. Huh. Yeah, that means there are a lot of spots with a lot of cells. Okay, let me just double check something. Okay, let's do a call sum on there's the total number of genes detected per spot. Yeah, for some reason, if you don't load matrix, it's going to think it's the other matrix, and it doesn't understand how to work with sparse matrices. Don't understand why it can't just infer that. Yeah. Anyways, that's why I was giving this error. Okay, so now lib size is this, the number of genes, total counts detected per spot. Okay, I'm gonna just plot the position colored by the library size. Oops. Oh, I see. The names of the positions has been erased. So let me just make sure I preserve the names. Yeah, okay. Pause is pause. .info 5 to 6, but the row names of the position is going to be the first column. Okay, now that looks better. Okay, maybe that's what was happening. Nope, nope, still looks funny. Never mind. Interesting. Maybe you have to log it. I'm surprised that there's such a high detection of stuff outside the tissue. Interesting. Well, still running. Okie dokie then. Something funky is going on. Okay, let's see. Yeah, let's try to figure out why is there so much stuff outside the tissue. I feel like this is not because of actual biology. I feel like there's a bug somewhere. Okay, let's see. Oops. We're double checking that this is the same dimensions. Yeah. And for summing up the columns, what I was expecting to see is that mostly there's a region that's going to be on the tissue that's going to be really high in the gene counts, and stuff outside the tissue that's not going to have such high gene counts. Does this function actually work? Looks reasonable. It's just getting the path, and then count matrix, read call names. It looks reasonable. That's right. Okay, I'm just going to try to do this myself. So I'm going to read data. Going to read the matrix at MTX. I don't think this is needed. Double check that matrix is loaded because I think read is a function of matrix. And yeah, now barcode is going to be something like this. You want to double check the dimensions. Yeah. Okie dokie. It was false. And gene info. It's going to be the features. The way header is also false. Oops. Now we need to separate by a tab. Okay. And so let's see. Barcode is just barcode.info1. I always like to look. Yeah, that looks good. And my gene is, let's make it the gene names. I'm just going to be column 2. And yeah, I should have actually double checked. I always like to double check the dimensions. Make sure everything is matching up. Yeah. Okay, so now I have a count matrix. Now let's see, is this any different? I could just try to say equals equals to that membrane raw, but I feel like large matrices doesn't like that. Okay, well, it looks the same, so I guess it is working as expected. Well, I'm still going to update everything to count that. Did this finish? Oh my God, it's a second iteration. Okay. Well, fine. We'll just continue with the STdeconvolve analysis on this kind of funky data set. Okay, maybe another sanity check. I'm not going to color by the total library size. Okay, we previously saw that certain genes are, very spatially defined. Just always good to sanity check. And so I'm going to look at the count matrix of this particular gene. Okay, that looks good. Oh, let's see. Okay. It's not in the same order, is it? Oops. Okay, I think I got it. Oops. Yeah. Okay. Yeah, this is why we should use spatial experiments and other objects. Will do the sanity checking for us. Okay. So the issue is the positions and the barcodes are not in the same order. Okay, so which do I want to change? Oh my God. Fix order. I'm gonna make positions the same order as the counts matrix. So call names, count net. Oops. Now everything's in the right order, so this should look more normal. Okay, much better. Oh, weird. Why is my brain rotated? Is that normal? That's gonna drive me insane. And okay. Why don't I? Okay, it needs to be rotated. Fix position. See, how do I rotate things? If I can just swap the X and a Y. Yeah, but then I would need to flip y. Well, I could do that. Okay, okay, yeah. So I like the way it looks here. Yeah, I don't like looking at brains upside down. Okay, so I'm gonna switch the x and the y, and that means the y position now needs to be inverted to we do a negative of it, it's going to be inverted, but that means, oops, let's double check. So yeah, now it's the right side up, but I think now the y positions are all negative, which kind of annoys me. So I'm going to just if you minus them in, then it should become positive again. Yeah, okay. And double check. Okay, thank God. That looks a lot better. And hopefully this, yeah, also makes more sense. So again, I'm just going to test a couple other genes. Well, my God, this is still running. Okay. Yeah. Okay, that looks reasonable. Yeah, I think this visualization makes a lot more sense to me than maybe it was just some of the color schemes. Yeah, I guess it's just the color scheme here. It's a little confusing. So I always interpret it as there's something here. Actually, no, there really isn't anything, which is why it's gray. Anyways, okay. So now let's actually do this STdeconvolve analysis. So I'm going to go back to, yeah, just regularly cleaning my matrix. And hopefully this will be more reasonable. We're basically filtering out the spots with too few genes and also the genes with too few spots. And I still want to filter out more things. Datasets, yeah, I want to basically get rid of this little chiplet there. So I think I should be reducing drastically the number of spots I have. So let's double check that. And then I want to just double check that my spots have been filtered. Doesn't seem to be filtering out the bad spots. Oops. Guess there are a lot of genes here. Weird. Okay, let me double check this clean counts function. The row sums need to be greater than min reads, which would be the genes in this case. Yeah, I want the call sums to get filtered. So the min lib size, well, I guess needs to be increased. Oh, okay. Let's double check. What's a reasonable filter? Yeah, these are probably swamped. So let's say 3.75. Let's see, what is 10 to the 3.75? Oops. Now 5,000. Let's see if that does anything. Basically, I'm expecting the spots outside the tissue to get filtered out through this cleaning. Yeah, in theory, I could have just downloaded the filtered matrix, but it should get me to the same place. Yeah, now there's a lot fewer. Oh, wow. This is a really deeply sequenced data set then. Maybe I should filter the genes really heavily too. Oops. Okay, and that means, yeah, again, sanity check. Okay, now this is more reasonable. Okay. Yeah, so, oops. Yeah, let's see, there's still quite a lot of genes. Again, this is probably a really deeply sequenced data set. Oops. Control C to stop the command. Let's try again. Okay, this looks much more unimodal. And much better. Now we have about 5,000 genes, 2,000 or so spots. Okay, this looks much better. Can even double check that my favorite genes are in the data set still. They haven't gotten filtered out. Yeah. Okay, that looks good to me. Okay, great. Now we can continue on with the rest of the STdeconvolve analysis, where we can filter for the corpus of highly variable genes, and also remove genes detected in all these spots. Yeah, let's check the dimensions of our corpus. It's just another counts matrix. This many genes, this many spots. Great. And I'm going to just choose the number of optimal cell types a priori. I don't want to do a whole search for it. Just going to guess around 25 or so. And yeah, that means that I guess if I pick the optimum of the min, it's going to be, there's only one. So all the optimums are going to be the same, but I can just pick the 25. This might take a few seconds. And this is still going. So yeah, once again, we are in the waiting realm. And this is where we start goofing off on Twitter and Reddit. Hopefully this won't take long. Oh, guess I'll check back and grab some water. Yes, I do code under a heated bucket. Looks like things are still going. I deeply regret doing this on a computer from 2013. It's an older computer. It has iMovie and their video editing software. Struggling real hard, struggling. Computer, not me. Oh my God. Okay. I think it's finally done. Okay, let's see. Yeah. Okay. Oh my God, it's finally done. Okay. Let's see. Yeah. Let's go back then to the vignette. Okay, we just decontaminated the data. Now I can visualize the decontaminated genes and see. Okay, I don't remember what that actually looked like originally. Right, so why don't we put them like next to each other? So the original data and now the decontaminated data. What does this function actually do? Okay. Boulder dash. Okay, how do I actually read the code? Not very familiar with these objects and okay, yeah, I don't know how to actually get to the code. So let's see if I try to find the SpotClean function. Very easy way for me to find it. Package. Oh my God. Yeah, send it somewhere here. Yeah, okay. Okay, not an argument checking. Okay, so this is what it's actually doing. So, okay, so it's, yeah, sliding along, estimating some contamination parameters. Oof, okay, this is, yeah, okay, so for me to actually understand this, I'll probably have to dig into the paper's methods. A little challenging to work out, but yeah, when you're calling this method, it's actually basically running this. Yeah, I wonder, how does the documentation work? Yeah, I'm not very familiar with these kind of particular ways of writing functions, calling other generics. Okay, but anyways, fine. Let's go back to, yeah, I guess in this analysis code stream, whatever, we're not really trying to understand what the method is doing. We're just trying to use it and kind of explore some of the computational components and kind of see whether it's worth taking into to understand further, maybe for a journal club. So let's visualize the original and compare that to the decontaminated. Okay, so it removed a lot of the stuff and okay, right, if I recall correctly, this is just a matrix. So what is the visualize heat map function? Oh my God. Okay, there's no way for me to actually read the function. Okay, my best guess is that it's, it's going to take in a matrix. So I could also just modify this original matrix to have the same sets of pixels, because right now the change in the pixels is kind of disorienting for me. It makes it hard for me to visually compare. So I'm going to subset to the same set of pixels. Oops. And double check. Yeah, okay, so I can subset to the same set of pixels. And now I'm going to visualize basically the original not decontaminated gene expression profiles, but restricted to the same set of pixels, the same set of spots. Yeah, that should make it a little easier to see what the difference is. Yeah, so this is the original, restricted to the same set of spots. And this is the new. Oh, okay. It's a little easier to see. You can see kind of the spots here have been kind of cleaned up. Yeah, actually, spots in the middle also are now a bit cleaner. Huh. The range of expression is very different. Interesting. Okay, so maybe I'm trying to understand how this has actually changed the gene expression. So why don't I look at kind of the original for this particular gene, so C78.7, and now the new is going to be that, right? So I'm taking the gene, and now this is going to be a vector. Let's double check my vectors are the same. Yeah, journal. Should be a vector. Okay. That's some kind of graph. I was hoping to just get the vector. Can I cast it to a vector? What is? Yeah, what is? Well, at least they're both summarized experiments objects. Yeah, it's one by number of spots. Because I want to see how do they stack up against each other. Yeah, so there's no way of plotting this as a summarized experiment object. I want to pull out the vectors. Okay, so yeah, I don't know actually how to cast summarized experiment objects into vectors. But there should be a way for me to get the, yeah, just pull out the data. By accessing the slots. Okay, great. Yeah, so this is actually what I want. Okay, I'm just going to call it X for the original and Y for the decont. Yeah, so yeah, this is, I'm getting the raw data stripped of all the kind of spatial experiment object stuff. And now, yeah, just double checking. Shoot. Okay, it looks like it's probably called some other name. Okay, let's try this again. I'm going to go into assays. There's data. Okay, it's called decont. Oh, shoot. Oh, right. It's called new. Yeah, I don't want the whole decont. I just want my one gene. Okay, so now this should be right. Okay, always good to sanity check. Yeah, so my y should also be a vector of the same length. Yep. And now I can plot x versus y, which will be the original versus the new decontaminated gene expression profiles for this one particular gene. Okay. Oops. Just going to remind myself, what is this gene? I'm going to change it to little dots. Yeah, so you can see, some things have shifted. The magnitude has shifted quite a bit for some reason. But yeah, overall, very correlated. And just always fun to do a core test. Doesn't like sparse. So let's cast it into a numeric. Yeah. So it's a super high correlation between these expressions before and after cleaning, which is good. It's not changing it too much. But yeah, definitely looks like, let's see, some things that had high expression before is now 0. Yeah, which is kind of what we would expect. And okay, always good to double check a second gene. Yeah, let's try the GAT2 again. Oh, actually, yeah, definitely try GAT2. And okay, it also looks, let's see, pretty similar in terms of the correlation. Original, not decontaminated. Yeah, you can see a bit of a change. Okay, cool. Last check, our last gene. Okay, again. Yeah, this is the original decontaminated. This one may be a little less dramatic. And again, we can quantify that a little more. Looking at our correlations. And yeah, a little bit lower expressed gene. Yeah, still very high correlation. Okay. Yeah, so let's see. So now the question from the student was to, can we combine this with STdeconvolve? Deconvolve. And that means that we're going to take the deconvolved matrix and use it with STdeconvolve. Okay. And yeah, it looks like the original results are still running. And again, this is a computer from 2013, so not the best choice in terms of computers for running, testing a code, code running thing. Anyways, yeah, so okay, so if we wanted to use it with STdeconvolve, say, what do we need? We need some type of counts matrix, which now will be the new, I guess, decontaminated gene expression. But again, we don't want the whole spatial experiment object thing. I just want the data. Yeah, I think this will do. Double check. Yeah, now it's now rather than dealing with the whole spatial summarized experiments object. I'm just dealing with a sparse matrix with that many genes and that many spots. And I think we should be able to just pipe it in. Why don't we give it a go? And actually, no, I'm going to do the same type of cleaning as I did before, just to keep things consistent, so that if there are any differences between these two results, we'll be able to figure out what was the difference. Except now here, we have this new CD. I'm going to call it 2. Surprise this one. Okay. Anyways, does it actually run? Oh, shoot. Yeah, let's load STdeconvolve. It works. Okay, whoops. Figure margins are too large. Oh, can't just replot it. Well, let's see. I was trying to plot the distributions. Okay, that looks pretty good. And now we can feature select again. Boop, 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 boop. Nope, counts 2. Corpus, no, we'll call it corpus 2. Okay, and again, I'm going to just avoid this whole searching for the optimal number of cell types and just set it to 25. Okay, so this is throwing us an error, and it says counts must contain integer gene counts, right? So in order to do latent duration allocation modeling, this corpus needs to be comprised of counts. And actually, I guess, this is a counts data set. This doesn't really make any sense to have, 0.1 of a gene. Yeah, so actually I never checked what are the values in this, I guess in this CD, right? Whoops, other way around. Yeah, so saying that, let's see, in this spot we have, 1.2 of this particular gene. Okay, so I guess I don't really know what that means, but maybe, for me, we should only have like 1 gene or 2 genes or 2 copies. I guess in theory, we could scale it up to, multiply by some large numbers. So then instead of 1.2, you would have 12. Let's see, what are the ranges of the column sums? So these are, there's some spots with only 200 or so, some spots with 100,000. Okay. So it's very variable. And that's perfectly fine. With plain duration allocation modeling, we can handle cells with large variations in the number of genes. They just all have to be counts. Okay, well, why don't I, what I'm going to do is, how do we get integer counts? I guess we could floor it. So, if you have 0.2 of a gene, It's like having 0. If you have 0.9, don't have like another copy of the whole gene. I guess in theory, I'm just theorizing, but yeah, why don't we do a floor? That will make all the points a lower number. You could also ceiling, or I guess also just round. But yeah, going to floor, and I guess yeah, leave it as an exercise for viewers and students to try round. I think if I floor it, then now it's going to be integers. Yeah, now it's integers. And now I can do the same type of cleaning as before. And now my corpus should also be counts. You can also just sanity check. Yeah, and let's see then. Why don't I put the LDA modeling here too? Oh shoot, should have named it 2. Just so that we can compare. And well, then let's see. Yeah, so now we have our 2 sets of LDA modeling running. And great. Now we have our 2 sets of LDA models running, and this computer is going to be working very hard to try to get us some results. And yeah, I guess we'll check back once it's done running. Okie dokie, and we're back. And I decided to go walk my dog and go eat. So now suddenly the sun is gone, but it's only like 5. But anyways, so let's see, where did we leave off? Right, so we previously had our STdeconvolve on the original regular accounts, and we deconvolved 25 topics. Let's see, or 25 cell types. Let's see what that looks like. Okie dokie. And yeah, so I'm just trying to, I'm pulling out the optimal model, and we only ran 1 model. So, and now we're going to get the deconvolved portions of the cell types and the spots and the deconvolved gene expression. And let's see, wonder if this will work. Nope. And oops, we need to give it the right position. Oh, right, we don't have an anode object here. So yeah, let's take a look at what is the decon prop is yeah, we have 2,490 spots or pixels and 25 topics, so 25 deconvolved cell types. And those are the estimated proportions of the cell types. So we just have to visualize it, the positions. Let's see, should have the same names as the deconvolved. The same set of spots. Yeah, no, it's the same set of spots. Previously, these were the unfiltered spots. Okay, let's see if this actually works. Okay. It's also complaining that the columns aren't the right name. So I'm just gonna make a new variable. Yeah, column. It wants the names to be X and Y. Not sure why that's entirely necessary. I guess it prevents it from being named other things. I don't know if this R variable is going to be too small. But anyways, okay, so yeah, and in parallel, we ran a STdeconvolve analysis on the preprocessed data from SpotClean, where we decontaminated some of the lateral diffusion. So we just ran STdeconvolved on those cleaned counts. So now let's see. Now we can probably run the same things. So, oops, left LDA2. Just going to again extract the 25 topics. And let's see. Don't need to do all this anymore. Two. Okay, so oops. Do I not have a position object? Oh no, I don't. Okay, in this version, I never saved it as a separate thing. Fine, I'll just copy this over. Because I think we previously only read it into the slide info. Now I wonder if I can just pull it out. Let's see. So I think, what is the class? Okay, it's a list. Oh, well, that makes it easy. What is slide? Okay, it's a data frame. Okay. And that's kind of like our previous info. And pause is going to be just the, guess what's the image row? The image call probably is what it is. The row names are going to be the barcodes. Let's just double check that. Is that indeed what we want? Yeah, that looks right. It's just a bunch of spots. Actually, now we can do the PPOS is the restricted the same set of spots. Let's double check that that looks right and it isn't rotated and flipped around or something. Whoops. Huh. A bunch of NAs. What are the row names of deconvolve 2? Oh, that's right. Oh, whoops. Oh, whoops. Let's see. Both here. The row names of the position matrix is going to be the barcodes. So now we can filter for the set of barcodes of the same names. Okay, that looks much better. Still has that rotation problem. Okay. I'm gonna just copy over the same fix from before. And now it should be rotated. Okay. Perfect. And I never noticed previously the cleaning of the counts seems to have filtered out a lot of stuff here. Did we have that previously? Oops. And okay, so these are our results from previously. Well, let me just double check. It looks like we have spots here. Oops. Yeah, so this is when I was previously concerned about, well, R is too small. This is the radius of the circles. Actually, what is the range of the spots? Now these are pretty big numbers, so I don't know what units they're in. Maybe they're pixels or microns. I'm not sure actually. So I'm going to set it at 100. It's the radius of the little circles. Yeah, if we knew what coordinates this was, it would be easier to guesstimate an appropriate R. I think if there were microns, then 100 microns radius would be quite large. Anyways, while that's plotting, let me just go back, and as I was concerned, well, not concerned, I just thought it was interesting that a lot of this was removed. Just want to make sure that we're comparing apples to apples later on. And anyways, well, so what is the dimensions of, yeah, so this is quite a few spots smaller. Let me double check. Yeah, we're missing quite a number of spots, and my guess is they're the ones corresponding to these. So well, at least that matches. So where did they get filtered out? Did they get filtered out at the clean count stage? So that's also a small number of spots. So already here. Okay, so they did get filtered out here. Oops. This is still smaller though. Yeah. So it seems like, let's see. Right, what was one thing? Okay. So it looks like the SpotClean filtered out a lot of spots. And yeah, I suppose STdeconvolve also, I tried to use the same filters for consistency, but it probably removed a little bit too much. In hindsight, yeah, if I were to do this again, I'd restrict to the same sets, same genes and same spots to begin with. But anyways, yeah, for now, I don't think it's going to be that big of a deal, but we're missing some information that probably should be there. That was all we have, the ability to deconvolve these spots, so we filter them out. Yeah. Okay, so yeah, the same thing goes here. And okay, in this case, the p-pause positions are different from here. So they might have been scaled properly using the scale factors JSON. You can try to confirm by reading through the code. So they read the format, they read the CSV file, And yeah, they seem to also incorporate the scaling factor. Yeah, so if they incorporate the scaling factor, does that mean it's in microns now? Anyways, so therefore these positions are in microns. I don't know what units these positions are in. So let's, yeah, but it's probably still going to require us to tinker with the R. So what range are the numbers? Somewhat in the hundreds, right? They're in the hundreds. So if, let's see. If this was a perfect grid of spots, which is actually what we had previously. So there were this many spots. Let's say it was like a square. Maybe there's around 70 spots this way. So 70 spots span 350 whatever units. And yes, yeah, 300 or so. So maybe each is going to have a diameter of 5. So, okay, I'm just going to guess that it's radius of 2.5. Let's see how close I am. Yike. And let's see. Oops. Looks like our previous results came up. And was, okay, great. Actually looks pretty good. I don't like these lines though. So yeah, is there a way for us to get rid of these lines? Oops. Let's read the documentation. And okay, there's a LWD setting. So, but yeah, 100 looks reasonable. So let's try to do the same thought exercise with the, yeah, this case again. So it's like 70 or something, right? And if we, oops, if we look at the range of the values, so it's 1500. There's 1500 and 70 spots have to span that. Well, yeah, so then that's the diameter divided by 2 is 100. Oh, that's close. Nice. So yeah, so I'm gonna, okay, yeah, I'm gonna get rid of the border line thing. And yeah, likewise, I should have done it here too. Although maybe it won't look so bad here. No, I'm just gonna stop it. And okay, let's see. Cool. Well, let's see. It looks like, I wonder why this is taking a tiny bit longer. Even though it's roughly the, I guess it's a tiny bit more spots. Suppose maybe they're bigger. But anyways, that looks like, yeah, looks pretty good. You get yeah, so this is the STdeconvolve results with the SpotClean preprocessing. And even just visually, we can see, yeah, each of these little pie charts are colored with the proportions of the deconvolved cell types. And yeah, we can see that, oh, look, here's the probably pyramidal cells. the hippocampus, dentate gyrus, cortical layers, and looks first class. So there's some structures that we recognize. And yeah, let's compare that with very similar results, it seems. Also still layer structures. Yeah, the colors don't necessarily match, so it's a little hard to see. Yeah. Let's see, how do I go about comparing these results? I guess they're in different RStudio sessions. I was impatient. Yeah, why don't I save so that I can load them all into the same session. So I have to save the decomprop, decomjuxt. Maybe I'll save the p+2. In this case, oh shoot, I should have named it p+2. Because actually they're slightly different. So yeah, so okay, I'm gonna save it as results 1. And here I'll save it as results 2. So right now we have the deconprop 2, the pawpaws 2. Nice. Okay, so now I'm gonna create a new file where I can, actually, where did I save this? Okay, yeah, it's in the main folder. So I'm gonna just load up the results 1 and load up the results 2 in the same session. Everything's here now. So I should be able to, let's see, plot some intensities. Yeah, I don't want to spend all the time visualizing those pie charts. Well, so I'm going to use this function. And yeah, so we have our decomprop and our ppas topic. How many topics are there? Oh, right, 25, because I said it that way. So let's see. Yeah, why don't I just visualize one topic? Size. Sure. Oops. Okay, I'm gonna get rid of this stroke. Okay, maybe I can't get rid of it completely. But okay, that looks pretty good. Yeah, and in contrast, we have the decomprop 2 from the results 2 and the p+2. But the topics aren't like in the same order or anything. That was interesting. It's like the cells lining the meninges. Is that what it is? But anyways, wonder if, yeah, so these deconvolve topics aren't going to be in the same order. I wonder if we can match them up somehow. So we have these deconvolved expression matrices in both cases. So if I just correlate them with each other, I guess, let's see. Yeah, okay, I'm just gonna double check their dimensions. So yeah, they don't have the same number of genes. So if I want to correlate every deconvolved topic with everyone else, and I'm going to need a shared set of genes. So let's see, I wonder how many genes are shared. Oops, not row names, column names. So yeah, there's 25 rows, 25 cell types. And yeah, 970 genes in the first case, 1,000 genes in the second case. Majority are shared. So now, if I just restrict to the shared genes, oops, right, take on just 2 shared genes. I think, can I just correlate these two? Is that going to work? See now. So this should be 25 by 25. Shoot. Okay, nope, that was wrong. Let me transpose these and then, okay, now it's 25 by 25. So we're basically looking at which deconvolved topics are transcriptionally matched between these two versions. Oops. And yeah, we can see that, oh, there's pretty good correspondence. I mean, some of the, like for example, the first deconvolved topic matches with the 21st deconvolved topic. So the first deconvolved topic in the STdeconvolve vanilla matches with the 21st in the SpotClean version. And then there are some cell types that are kind of correlated with each other, both within, which is understandable since, you know, you're going to have neuronal cell types that are related and glial cell types that are, you know, related to each other or transcriptionally quite similar to each other. So that's not too surprising, but it seems like there's some correspondence. And let's see, why don't we just try to pick, yeah, so pick a match. So let's see, 1 and 21. So if I'm reading this correctly, it would be, this is STdeconvolve vanilla, topic 1, STdeconvolve SpotClean, topic 21. And these should look about, I guess I would hypothesize that the SpotClean version would be like cleaner version, maybe like a tighter, less diffuse kind of version. Let's see if that holds true. Okay, yeah, so we see some layer structures and okay, that's definitely not right. Did I swap the two? I always forget which corresponds to the row and which corresponds to the column. Okay, maybe it's the other way around. Let's see. It's the other way around. Hooray, it's the other way around. Okay. Good debugging. And yeah, so let's look at these two results. As originally, you know, is my hypothesis right? So in the STdeconvolve vanilla, you know, these are the estimated cell type proportions in the spots. You can see there are some spots with high proportion of this cell type and other spots with, you know, no proportion of this cell type. And let's see. Yeah, so I think my original hypothesis was that with SpotClean, you know, maybe some of these spots with kind of a low proportion of this cell type would kind of get removed because maybe the gene expression there is actually from the diffusion of the spots, cells in the spots here. But let's see. Well, it doesn't look like, I guess it does look maybe a little bit cleaned up, maybe. Okay, again, some of the spots seem to be, removed from the SpotClean. So the SpotClean removed some of the spots that weren't on the tissue, I guess, but that weren't registered well to the tissue. So it's a different number of spots. Okay, so that makes it a little hard for me to see. Maybe we should restrict to the same set of spots. So this would be intersecting the row names of PPOS and row names of PPOS2. So now I can see spot shared and do that also over here. I think I would also want to restrict the decontaminated to the same. Just move it over. Okay, let's see now what does it look like? Okay, now we can toggle back and forth a little bit better. Oh, look at that. Oh, yeah, it does. Tiny bit shifted with the, with the SpotClean. It's, like if you focus on like this spot, the deconvolve proportion. Okay, maybe on these spots, the deconvolve proportion does decrease, and the deconvolve proportions here increases. Oh, look at that. Huh. Okay, so well, that's just visually assessing. Why don't we do it a bit more quantitatively, the same way we looked at the genes previously? In the SpotClean. So we can always look at how the gene expression has changed from the cleaning, which is kind of at the molecular level. What does SpotClean do? But in this case, going kind of one step further, just because the molecular profile has changed a tiny bit, does that actually impact the downstream deconvolution? And how does the deconvolved cell type proportions change? And so I'm going to focus on this particular deconvolved cell type. So again, cell type 21 in the vanilla version and the cell type 1 in the SpotCleaned version, restricting to the shared spots. And I'm just gonna copy this over. And double check that it's, the length is the number of shared spots. And, look at that. It's pretty correlated, but definitely slightly shifted. So, 0.99 correlation. But overall, it's not drastically, dramatically changing our results, which is probably good. But it definitely has changed things a little bit, and perhaps is giving a result a bit more consistent with the underlying histology, something for us to kind of look at and check out. But overall, definitely promising. I think, there definitely is a potential way to, especially if you have a sample that has a lot of lateral diffusion because of sample processing. I think in this case, as you've seen on the molecular level, there wasn't that much of a change. So I wouldn't expect the deconvolved cell type proportion to be drastically impacted either. And indeed, it hasn't really been, but the trends are consistent with what we would expect. I think it's a reasonable preprocessing that is worth exploring. Again, if you have a sample that or a tissue that has a lot of anticipated lateral diffusion issues. Cool. Yeah, so I'm just going to save these results. And let's see. Now, why don't I think if I don't push these results somewhere, they're going to be lost forever. So I am just going to create a new GitHub repo, and the computer I don't typically use. Otherwise, I would typically be logged on. And yeah, let's create a new repo. And I'm just going to call it coding videos. Maybe I'll change it later. I think I would want to use this repo for a large code dump. And yeah, so actually, why don't I... Yeah, I kind of just want to commit everything, I think, for now. I'll also change this to coding videos. And just want to push all this code to GitHub or else I'm going to lose it. Okay, so I'm just gonna copy the commands. I'm gonna initialize Git and Git status. Yeah, I always like to add a git ignore. I don't want to commit my R data. I don't want to commit my temp Emacs files. The other stuff that I don't want to commit. Let's see. Yeah, I don't want to commit these rproj things like this thing. So I feel like there should be a template or something that I should just copy as a git ignore. Okay, so I do want to add my code. Do I want to add the data? Oops. Yeah, I don't actually want to add the data either, because this is something you can download. So it's a lot of stuff to push that's not necessary for reproducing this. Yeah, I'll just link in the README. Data from 10x, and I think that should be everything. Yeah, that's everything I want to save. And yeah, I can just git commit message, STdeconvolve with SpotClean, convolve with SpotClean, code demo. And yeah, I'm gonna branch and push to main. And it should be all set. Oh, great. Yeah, so that's how I would go about, doing exploratory spatial transcriptomics data analysis. I hope was educational, and yeah, thanks for tuning in.